last but certainly not least, we have our last speaker for before we take a short break, Dr. Dolores DeVizio. And while Dolores sets up, and she's on mute, by the way, Dolores, I'm going to introduce you. So Dr. DeVizio is a pathologist by training and a molecular and cell biologist who trained at Albert Einstein College of Medicine and Harvard Med School. She's currently holds faculty at Harvard and also has an academic appointment. Her lab is at Cedars-Sinai, which is in Los Angeles, and she is an associate professor. Dr. DeVizio is one of the perennial researchers who studies these oncosomes that Peter mentioned and what I mentioned, and these are those particles that are shed by cancer cells. Her team currently studies those at what we call EVs at Cedars-Sinai, and she has, she's really, one of the top researchers, and I really look forward to hearing what you have to say. So Dolores, the floor is yours.
Thanks, Josh, for the nice introduction, and thank you and the other organizers for putting together this panel that I'm very excited to be part of. So as mentioned already by Danny, by Peter, by Josh, there are these extracellular vesicles that are membrane-derived vesicles released by any cell in the body. And I will give you a very brief introduction mentioning that the importance of these vesicles is that they are released by certain cells and they can be taken up by other cells that are influenced by these vesicles. And if we imagine a cancer cell in particular, we know that there is a wide range of extracellular vesicles that can be released by this cell. And they can be taken up by all cells in the tumor microenvironment that are affected and reprogrammed in a way that ultimately promotes the cancer progression to metastasis. And I like to use this slide, this schematic, that really depicts the heterogeneity. So not only the cancer cells are heterogeneous, but also the vesicles deriving from these cells are highly heterogeneous. And while most studies historically have focused on very tiny vesicles known as exosomes that originate by a distinct pathway that involves the endocytotic machinery, the formation of multivesicular bodies and the fusion of these bodies with the plasma membrane for the release of the vesicles. We now know that there are other type of vesicles that originate directly from the plasma membrane of the cells by direct budding, and several of these vesicles have been already reported, and this includes the large oncosomes, which are significantly larger than the exosomes. And we were the first to report the presence of these vesicles, specifically from highly aggressive cancer cells that transition from a mesenchymal type of motility to an amoeboid type of motility that Danny has mentioned at the very beginning. And so we demonstrated that the loss or the silencing of cytoskeletal regulator DIAF3 induces the transition of the cancer cells from the mesenchymal to the amoeboid phenotype and results in the shedding of these very large extracellular vesicles. We found them particularly interesting because when we started looking at patients, and in this case we worked on prostate cancer patients, we noticed by looking both in the tissues and in the plasma of these patients that there was a significant correlation between the presence of these large vesicles and progression to metastatic disease. In this case, we have castration-resistant prostate cancer, which is metastatic. We also used some animal models that basically gave us the same result. When we look at particles larger than 1 micron, which is the case for the large oncosomes, we have a significant correlation with metastasis. And this is not the case if we look at the exosome-like particles because we know that all cells in the body release these small particles, not only the cancer cells. So a part of my laboratory is interested in trying to use these particles to develop a novel approach to liquid biopsy that Peter has already been talking about in depth. So I'm not gonna go too deep in this talk on the liquid biopsy approach. However, one advantage of these vesicles is that they can be found in the circulation in the blood of patients with any type of tumor, not just with prostate or breast cancer. And they can be, once isolated with specific procedures, they can be analyzed at the protein, DNA or RNA level. In fact, one of the advantages of using these vesicles as a source of biomarkers in the liquid biopsy approach is that we can look at different type of molecules that are enclosed in these vesicles, from DNA to RNA, but also proteins and lipids that all seem to play important functions in these vesicles. So we published a few years ago a paper demonstrating that when we compare the abundance of RNA in large oncosomes versus exosomes, we find significantly more abundant RNA in these large vesicles. The profile of the RNA in these vesicles is more similar to the profile of the originating cells versus the profile of the exosomes. We were able to do a whole transcriptome analysis using an Illumina ISIC 2000 platform at long time ago, and we were able to demonstrate that there is a significant number of reads of these messenger RNA transcripts in large oncosomes more than in the exosomes. And when we compare the profile of the RNA in exosomes and large oncosomes, we found a very striking similarity. So both vesicles, if they are derived from the cancer cell, contain similar transcripts with some differences that we are now analyzing more deeply. We also saw that the profile of the exosomes and large oncosomes is different from the cells of origin. And I think this is a very important point that is in favor of using these vesicles for the liquid biopsy, because cancer cells can shed material that is not necessarily highly expressed in the cells themselves. So we can use the blood to basically identify altered molecules from the cancer that the cancer cells are spitting out basically during the progression. We also demonstrated by using, by doing an in silico analysis using transcriptome data from the ENCODE cell line compendium that has specific analysis of the cytosol, nuclear, and all cell, that the profile of RNA in these large oncosomes is more similar to the profile of the RNA in the cytosol versus the nucleus. Which is important because it can help us understand the origin of these RNA in the vesicles. We also found that there is a significant enrichment in the vesicles in comparison to the cells of origin for transcripts that encode for transcription factors that are important in promoting cell division and inducing a transition from the G1 to the S phase of the cell cycle. And we also found when we looked at the patient plasma. So we isolated these vesicles also from the patients from the plasma of 10 patients with breast cancer. We found that these transcription factors are significantly upregulated in the vesicles from the patient plasma in comparison to the controls. And finally, again, we were very lucky to extract these vesicles from patients with breast cancer. We found that when we looked at all the altered transcripts in comparison to the controls in patients, we found, and we compared them to known transcripts that are upregulated or downregulated in tissues from patients with breast cancer. We found a significant correlation between the blood and the tissue, suggesting that we can use these small vesicles as a readout and as a non-invasive biopsy that can be potentially used also for monitoring. So I just want to reconnect very rapidly to Dr. Wen talk, where she nicely showed all the data on the PDX and using the PDX as a monitor of the response to chemotherapeutic agents. Obviously, the extracellular vesicles could also be investigated in those mice or in other models to basically have an idea in real time and without any invasive biopsy of the response of the tumor. I also want to mention another study that we published a couple of years ago and some unpublished data following that up where we demonstrated that large oncosomes are the vesicles in the plasma that contain most of the DNA. So they are suitable for genomic analysis. And we have now ongoing experiments where we are comparing the results of these liquid biopsy using large oncosomes versus circulating DNA. And what we are finding is that if we use very deep sequencing of all exome sequencing in the blood of patients, we can identify deletions and amplifications or copy number variations that are typical of the tumor, in this case prostate cancer, and can be easily identified in patient plasma in real time. We also can simply look at the abundance of these particles by using tunable resistive pulse sensing, which is a new technology that allows to quantify and measure the size of these vesicles. And when we look at the vesicles larger than 1 micron, as is the case of the large oncosomes, we can see that the abundance of these particles in patients, and we have here 4 patients with lung cancer, is significantly higher than in the control individuals. So we can easily, I think we are now ready to move to the clinics, and my lab is trying to develop clinical tools that are easily to implement in a CLIA lab. So regarding the function of these vesicles, which I think is also very interesting, what we have shown a few years ago is that large oncosomes, but not exosomes, isolated from plasma of patients with metastases, are enriched with a very important and active kinase in cancer, which is phospho-AKT. So we can easily identify this protein, in this functional protein in the vesicles. When we use large oncosomes to condition cells in the tumor microenvironment, as Josh was showing with all of our tools of cell culturing and transwell assays, etc., we show that the large oncosomes, but not the exosomes, induce activation of one of the most important oncogenes, which is MYC. And that this is dependent on the presence of the kinase in the oncosomes. In fact, when we inhibit the kinase, we see this phenomenon disappear. We also show that large oncosomes can induce expression of alpha-smooth muscle actin, which is a marker of reactive stroma, which is an indication of progressive cancer. and this is inhibited by inhibition of MYC. These vesicles can also induce angiogenesis, as shown by specific in vitro assay, and we can also inhibit this phenomenon by inhibiting MYC in the stroma. And finally, when we also did a mouse experiment in which we injected the cancer cells with the stromal cells together, and the pre-treatment of the stromal cells with large oncosomes significantly increased tumor growth. And when we inhibited MYC, there was a complete suppression of these phenomenon. We are now moving forward to a model that is more representative of metastasis, because we know that both in prostate and breast cancer, metastasis can occur several years, if not decades, after the primary tumor. So we're now using bone marrow mesenchymal stem cells, and these are primary cells isolated directly from human individuals. And what we, so after characterizing our vesicles using rigorous methodologies, we have basically shown that if we treat these cells with exosomes and large oncosomes, we can see a strong upregulation of mRNAs codifying for chemokines and cytokines that are very important in tumor metastasis. And this is a dose-dependent effect that is significantly more powerful when we treat the bone cells with oncosomes in comparison with exosomes. We have now performed large-scale analysis in this bone marrow mesenchymal stem cells treated with large oncosome and exosomes, and we show that the number of genes that is influenced by large oncosomes is significantly higher than those influenced by exosomes. And we have also shown that so this is also evident here in these heat maps that show all the genes that are influenced by exosomes here and large oncosomes here. And as you can see, the signal is much stronger if you look at the intensity of the red and the blue in large oncosomes treated cells than exosomes treated cells. And if we look at the top 50 genes that are upregulated and the lowest 25 genes that are downregulated by these vesicles, we find that the most important alterations have to do with the most important pathways that are altered by these vesicles have to do with neutrophil migration, with cell chemotaxis, and with an inflammatory response. So as Danny had mentioned in the introduction, the tumor microenvironment is very important in the response to the tumor, and we are really understanding more and more how different type of immune cells can respond differently to the tumors in ways that probably initially try to block the tumor, but then eventually result in promoting tumor progression as well. So we have done experiments where we show that both exosomes and large oncosomes can induce, when they are used to treat the bone marrow mesenchymal stem cells, can induce migration of neutrophils and cancer cells, suggesting that they might be recruiting these cells to the bone to basically generate what is known as the pre-metastatic niche. So in conclusion, what I've shown you today is that extracellular vesicles are not just exosomes, but they can come in different sizes. They have distinct cargo molecules in them, and they can play distinct functions. The large oncosomes are cancer specific, and they contain cancer-derived molecules that can be interrogated in patient blood. Large oncosomes can reprogram the cells in the tumor microenvironment, inducing an inflammatory response and other modifications that are pro-metastatic. Of course, we need additional investigation to really understand how these distinct pathways can be altered by distinct vesicles, because one of the ultimate goals of our research is also to try to understand how these large oncosomes are formed and how we can block them, because obviously by just blocking exosomes, we're not going to block all the intercellular communication that occurs between cancer cells and the tumor microenvironment. And I want to thank first of all the students and postdocs in the lab who performed the actual work, and several collaborators that have been very helpful in these past 10 years, the funding agency, and obviously the patients for sharing their specimens. And thank you for your attention.
Thank you, Dolores. That was great and fascinating.